record on this computer so I can post it for y'all. I'm going to make that big. If y'all notice that anyone's in the waiting room, can you just let me know? Because sometimes I don't see that. Okay, we're recording. Okay, so this exam is going to cover chapter 20, 21, 22, 23, and 26. And hopefully y'all saw on the schedule that I provided that we're not going to cover every single section of those chapters. So while it sounds like it's a lot of material, it's really bits and pieces from each chapter. So it's not hopefully as overwhelming or as much info. This exam has more chapters and information than any of the other exams that we're going to have. I couldn't break it up any differently, so it ended up being this way. So chapter 20 is all about phylogeny, which is the way that we figure out the evolutionary history of species and how we can use phylogenetic trees to determine where common ancestors are, who is more closely related to us or certain species, and why that is. And it's always because of a common ancestor. And then systemics is classifying those organisms to determine their evolutionary relationships. Taxonomy is what humans do because we like to organize things. And so taxonomy is the term that we use to organize things and name those different organisms. And if I'm right, I don't know if I want to get into this just yet. Maybe I do. So one of the terms that you'll hear is like a common ancestor. And a common ancestor, like this ancestral lizard, is no longer in existence anymore. And all of these guys are what has derived from that common ancestor. And wherever you have these branches in these lines, these are where there was an extinct common ancestor. So if you're looking at this phylogenetic tree, where you have the little branches like this where they share a common ancestor, these two guys are much more closely related than even iguanas and geckos, even though they look very similar, or this monitor lizard and the iguana, because their common ancestor was a little bit further behind. So you might get like an art question or a scenario question where you're shown a phylogenetic tree and you might be asked which pair is the most closely related, and you would just go kind of, if it was like, if your option was monitor lizard and who else, it would have to be iguana because their common ancestor is more closely related or sooner in the phylogenetic tree than with snakes or geckos. Oh, I'm going to clear that. Okay. And this is just another phylogenetic tree, kind of showing the same thing, the rise of Homo sapiens. So, you see this all the time, and this is really not an accurate depiction of what arose from humans. These are extinct common ancestors, but we didn't really start to stand up straight and all of a sudden become a human. There were steps in between where things went extinct and gave rise to brand new species. So it's not necessarily a gradual progression like this image kind of depicts. It's much longer, and it takes years and years and years, millions of years. So here we're up to like 6 million years ago. We had a common ancestor with every single animal that you see on here, and we're not very closely related to chimps because our most closely related common ancestor was down here, right? Because that's where we branch off separately. Sorry, just making sure nobody is in the waiting room really quick. So we're more closely related to Homo erectus and the Neanderthals than we are ever related to chimps. Chimps still exist, Homo sapiens, humans still exist, and our common ancestor that we diverged from to become separate species is gone, no longer existing. Clear that. Close. Okay. So I would know about binomial nomenclature. This was kind of developed by Carl Linnaeus. He is the father of natural selection and evolution, and I'm sure you've heard his name before. He discovered the Galapagos Islands, and that's a very unique ecosystem because it is its own little island and separate from everything, but small enough that he was able to identify all the different species that are on there. So we'll talk about Carl Linnaeus later on. But this binomial system is a two-part system. So Homo sapien is our scientific name. The first part of the name is the genus. There are many other homos, right? Like you saw on that image that I showed you earlier. So that's kind of like your first name. There's a lot of Lucys or Paulas, but your last name makes you a very specific person. And so that the last part of the binomial structure is species. Genus is always capitalized, species is not, and it's always either italicized or underlined. So I wouldn't be surprised if there was a question on there where it gave you Homo sapien in like 4 different ways and asked what is the proper way for binomial nomenclature. I would also know the hierarchical classification. So you should remember this from the first part of biology 1406. And it's basically just the taxonomic groups. And so it goes from the way that it's written right here is from like very specific to more broad. In my brain, for me, it's much easier to think of it as an umbrella. Like a big umbrella is the domain that everything is underneath. And then you have smaller umbrellas. Kingdom is a little bit smaller umbrella. Phylum is a little bit smaller than that. Class, order, family, and then genus and species is incredibly specific. So this is an upside down thing, but domain are these giant things that encompass all sorts of stuff. And then when you get into kingdom, we've now separated out protists from eukaryotes. Kingdom Animalia is all the animals in the world. Phylum Chordata is a little bit smaller, and so we've separated out anything that doesn't have a ventral nervous cord. And then class Mammalia, no longer are we looking at fish and reptiles, amphibians and birds. And then it gets thinner, more and more and more confined and restricted as you go on. So I kind of already mentioned how you kind of read a phylogenetic tree. So each branch point, which were those common ancestors where it splits off, that's where you diverge into 2 different groups of species or individual species. And then sister taxa are groups that share a common ancestor. Let's get through these terms and then we'll look at it, I guess, a little closer. So rooted tree is a branch that represents the most common ancestor of all the taxa in the tree. Basal taxon, it diverged early in the history of the group and it's kind of all by itself. That's what, when we were looking at the Homo sapien, that's what the chimp was. It's all by itself. And then polytomy is when you have more than 2 groups emerging from a common ancestor. So one is your ancestral lineage. And here we've got these branches off. This little guy all by himself, there's nothing else with him. He branched off early and he's still alive. That's a basal taxon. The sister taxa are like B and C because they share a common ancestor with nobody else. Branch points are all of these 1, 2, 3, 4. These are all extinct common ancestors to all these. So D, E, and F are also technically sister taxa, right? Because they share number 5 as their common ancestor. Does that make sense to you guys about how to read that phylogenetic tree? I don't want to repeat it over and over if you're like, I got it. Okay. So phylogenetic trees really show you history, but they don't show you much more than that. So they don't really show how similar you are in the way that you look. It just shows where you came from. So there can be 2, just like with the lizards, that one lizard looked a lot more like a snake, that legless lizard did, than its very close common ancestor. So it's not necessarily how similar you look to someone, it's what you arose from. We usually use DNA to figure it out. I just want to go through some of these kind of quickly. Okay, so I think in 1406 this was covered, but the difference, just to give you a review, the difference between your phenotype and your genotype are two different things. Your genotype is what's in your genes, what your genes have on them, and they tell your body, or they tell you what to look like, right? And your phenotype is how you look. So if you're talking about the common example used all the time are peas. If you do have like colored peas, yellow peas, or green peas, the color is their phenotype. A gene makes it be that color, or wrinkled or smooth peas. So that's the phenotype. It's the way, it's the characteristic of the way that they appear and look. And when you have similar phenotypes and genetic similarities, we call those homologies. There are homologies and there are analogies. And a homology is a similarity you have because of a common ancestor. An analogy is something that you have because you just evolved separately and differently and have similar traits, but not because you're related in any way. So, and that's due to something that we call convergent evolution. So the North American mole and the Australian mole, they are genetically similar, related organisms, and they share similar traits, the big one being their feet and their little short tail so they can dig. So that is a homology because it's a shared similar trait due to a common ancestor, even though they are anywhere near each other, anywhere in the world. Well, they're not near each other because one's in North America, one's in Australia. A very kind of quintessential example of an analogy would be wings of birds, bats, and or butterflies, right? So butterflies have wings, but there's no bones in them. They just developed and evolved wings at some point. Bats did as well, but they're not analogous. They're not homologous, they're analogous. Instead, the homology is with our hands. That's the bat's wings. May not do the same traits or may not do the same function, but looking at it, the bone structure is the same. It's part of the bottom of the arm, that kind of thing. So all of these different, they're not all mammals, but all these different chordates, I guess. Just pretend they're mammals and just don't look at the bird. But we all have highlighted in yellow are analogous features that are like the tips of their little fingers. And you would not say that, a hand is analogous to a wing, but you could say these two wings are homologous. They've evolved separately. So that's the difference between an analogy and a homology. Cladistics, this classifies organisms by common descent. We've been looking at this when we've been looking at the phylogenetic trees. So there's different types of clades. A clade is a group of species that includes the ancestral species, the dead one, and all of its descendants after that. And so there's a monophyletic clade, paraphyletic, and polyphyletic. Monophyletic means that it consists of the ancestor species and all of its descendants. Paraphyletic consists of an ancestral species and some, but not every single descendant. And polyphyletic has various taxa. So, I forgot to delete my old notes in here. So in a monophyletic clade, this would be group 1, which includes the ancestral species and all of the common ancestor, all of their descendants. A paraphyletic is group 2. And in this case, we're just looking at D, E, and F, but not G. So it includes some, but not all. And polyphyletic will include the common ancestor plus someone else's common ancestor over here. There's still notes in there. So what is this trying to show us? So our common ancestor is this guy over here, but this paraphyletic group is, look, this green or blue, kind of highlighted shaded section is paraphyletic because it includes some plus some others. this one is a polyphyletic, right? Because it includes from multiple different, actually, I'm sorry, this doesn't include these guys. I have to not look at my purple part. Polyphyletic is yellow because it's including the seals and the cetaceans. The common ancestor is way over here. So it's taking two different clades, because technically this is the common ancestor and this one is for seals and bears. Isn't that crazy that seals and bears are more closely related than cetaceans, which I guess are dolphins? Maybe y'all know that better than I do. And this one's paraphyletic because it includes some, but not all. If it was all, we would also shade all the way down to the cetaceans. A shared ancestral character is a character that originated from your ancestor, and then a derived one is just unique to that clade. So in this case, the unique or the shared derived character would be hair, because our common ancestor didn't have it. But probably having four limbs is the shared ancestral character, because the ancestor has four limbs and so do all of the descendants. So remember that we talked about a basal taxon being kind of the thing that's out here. It's also called an outgroup. So an outgroup is a species or group of species that's closely related to the in-group that's being studied. You could also say that this is a basal taxon because it derived early on and it's still alive. Okay. Are y'all good on understanding how to look at those phylogenetic trees? Because that's kind of that is the main thing, along with some of those, lots of those terms.
I guess the only question, and I guess maybe because I didn't really understand it too much, was the maximum parsimony.
Maximum parsimony.
Parsimony. Yeah. I was trying to kind of
Parsimony, sorry.
yeah.
Let me get back in here.
Kind of grasp it.
Wish I had.
So that's because I saw from the notes and it says, so would it be like something that would have the same derived character? They would share the same characters, but then I guess they would be like in a smaller, I guess.
It's just a term, or it's not a theory,
Okay.
but not a belief, but it's a term used. So when you develop a phylogenetic tree, there are scientists can argue and say, well, this probably isn't totally correct. It should be something more like this. When they use maximum parsimony, they're trying to choose the fewest evolutionary events. So the fewest branches off of that. So there's probably a lot of things that we think are closely related that may not necessarily be. It's kind of like, the simplest answer is usually the right one, right? The simplest idea is generally the right answer, or simplest explanation is that's kind of what maximum parsimony means, that the fewest evolutionary events is the most likely to be the result of what we see in these organisms that we're looking at. Okay. Whoops. Okay, I stopped sharing. Now, I think chapter 21 might be one of the more confusing ones. Let me see if I can find it. 21. Because this one is about evolution and Hardy-Weinberg, and I don't know if this one talks about bottleneck or if that's in chapter 22. I can't really remember. But it's a major misconception. People always think that organisms evolved during their lifetimes. An organism cannot evolve. Populations evolve. A population is the smallest unit that is able to evolve. We adapt. Individuals adapt, but we don't technically evolve. And evolution is a change in your genetic code, almost a total change in the way that you look. And an adaptation would be you are a bear that grows really fuzzy hair in the wintertime, but you haven't changed your insides or your genetics. And so like a really good example of evolution and natural selection is something that happens because of evolution. It's a way to explain evolution, but probably the most common explanation for evolution is beaks with the Galapagos Islands and what Carl Linnaeus found. What Charles Darwin found. I think I told you earlier that Carl Linnaeus did Galapagos Islands, but that's not right. That's a different guy, Charles Darwin. But he was able to find that there were certain birds that lived in certain parts of this island or a certain island where there wasn't a whole lot of insects, and they mainly fed on berries. And so their beaks evolved to be able to crush the nuts that they were feeding on, versus other birds that nested on the ground and they fed on things in the soil. And so now they have a different type of a beak to dig in the soil to grab those worms. Or if you look at a toucan that eats whatever it is that toucans eat, their weird beak has evolved over time to populations have evolved to have that really strong beak to crush, I'm assuming, nuts and wood and stuff to get whatever food it goes after. And so in this example of these finches, the average beak depth increased, and I don't know what depth would be on a finch. I'm assuming it's like how big it is this way, not how long it is. But it increased in the finch population because the birds with the smaller beaks were less likely to survive a drought. And so over time, and this was 3 years, I guess, when you measured all of the beaks of the finches, the populations now had larger beaks because they were able to probably crush things that had some moisture inside of them. And so the population evolved, but it wouldn't have happened in the lifetime of that one individual bird. What would happen is that all the birds with little beaks that died of the drought would die off, and their genetics no longer transferred, and they weren't able to produce offspring that would live. Instead, finches with larger beaks survived, mated, produced other finches with larger beaks, and they just kept getting larger and larger as time went on. So microevolution is a change in allele frequency in populations over generations. Hold on, am I on the right chapter? We did 21 and we did 20. So microevolution is a change in allele frequency in population over generations. And this can happen 3 ways: natural selection, genetic drift, and gene flow. So alleles, I think we're going to look at this a little bit more, but alleles are like individual genes. And you either have a dominant allele or a recessive allele, right? Yeah, that's just the intro, and it kind of happens too fast. It's a little confusing. So 21, we're only covering sections 2 and 3. We have to have genetic variation in populations, otherwise it wouldn't work properly, right? If we don't have genetic variation, then if we have a cold snap like this and there's no genetic variation in a certain bird population or squirrel population, some of them aren't fat enough to be able to survive that cold weather, or some of them aren't large enough to be able to produce enough heat, and everybody dies off. So by having genetic variation, you are able to have sustainable populations that can survive varying environmental and other types of conditions. So a population is considered a localized group of individuals that are able to interbreed with one another. And populations can be isolated in location, but not always necessarily the case. So like in this example, there's these herds of, y'all guess what they are, moose or caribou, I guess is what they look like. Genetically, they're very similar, but they're technically 2 different populations. And although they overlap, for whatever reason, they don't interbreed with one another. So they're 2 separate individual populations. This seems out of order a little bit, this slide, but a gene pool is all the alleles for all the loci on your chromosomes in a population. Loci or locus is the location that the allele is found. An allele is a section of that gene. So when you think of genes, it's usually your whole chromosome made up of tons of genes inside of that. Well, we have 26 different 23 different chromosomes, right? Chromosome 1 might tell us all of the color and pigment in your body, and also if you're going to be tall or short, right? And there's tons of different things that make us who we are and make us all very genetically independent and different. And the allele is what tells you your hair color, maybe. And there's probably multiple alleles that give you the different coloring for hair. And then there's even other alleles that say you're going to go bald soon, or you're going to always keep your hair, or you're going to have gray hair, or you will never have gray hair. So there's many, many different alleles that act on our body and give us our different phenotypes throughout our life. And where they're located is called the locus or the loci, because you have two of them, right? So locus is the location that defines it. We have two chromosomes, one from mom, one from dad. And so whatever the allele or gene is that we're looking at, there's actually two locations for it on the two different chromosomes. So the frequency of an allele in a population can be calculated, and that's the Hardy-Weinberg equation. So we can look at a population and know about how many people or individuals are in that population, and we can determine what is the percentage of dominant, homozygous dominant, or heterozygous or homozygous recessive. Remember, homozygous dominant, homozygous, homo means same. So it's that you have got the same for mom and dad. Heterozygous, hetero means different. So you got a different gene from mom and a different than what dad gave you. And homozygous recessive gene doesn't show up if the dominant gene is there. So, sorry, I was just checking who was coming in the door. So homozygous dominant means you got the dominant pair from both mom and dad. Recessive, you got the recessive set from mom and dad. And so you can figure out the frequencies of alleles, and the way that you do that is by this little equation called P plus Q equals 1. The frequency is like the percentage, right? And if you add it up, it should be 100%. The frequency of how many of a recessive allele or a dominant allele is in the population should be, when you add it up, should be 100%, because you don't have any other options. There's not a third gene that you could get. It's either dominant or recessive. So if you look at this population of these flowers, if you are homozygous dominant, you have both dominant genes from mom and dad, you're a red flower. If you are homozygous recessive, you got both recessive genes from mom and one from dad, you are a white flower. And if you're heterozygous, you are a pink colored flower. So you got the dominant, you got a muted version of the red version. And if we have a population, if you add up these 320 plus 160 plus 20 should be 500, right? So we have 500 flowers in this population. We can, I'm too ahead. So we can figure out, I don't want to look at this one first. Well, let's just do the P plus Q equals 1. Oh, I don't like that either. Hold on a second. Oh, I know why we're doing this here. So this is CR, the red is the dominant allele. Well, we have 320 red flowers in our population, but we also have two of those alleles, right? Because it's homozygous. So 320 times two of those alleles, plus our heterozygous. We have 160 pink flowers, which means we have 160 red and 160 white. So that's where that 160 comes from. That leaves 800. That should add up to 800. And then our recessive allele, we have 20 white flowers. So 20 from mom and 20 from dad, 20 times 2, plus the 160 of those heterozygous. That's one way to do it. The way that I always learned to do it Well, actually my way is a little bit more complicated. That's a little bit easier. So we're able to figure out what our P and our Q are. And I know P is usually your dominant allele and Q is designated for your recessive allele. So P should be 8. If the total alleles is 1,000, right? P should be 800 divided by 1,000, which should be 80%. So 80% of this population of flowers in your garden bed have 80% of the alleles are red. And then white, 200 divided by 1,000 should be 20%. So 20% of those flowers have the recessive allele. So we have our P and our Q, which is what I did right there. And the sum should always equal 1. If double check your math, if you have to do this math question to figure out P and Q, if it doesn't add up to 1, it's not correct because it has to be 100% in the end. So the Hardy-Weinberg equation describes the genetic makeup expected for a population that is not evolving. So there's nothing occurring, there's no evolution happening. And the Hardy-Weinberg equation is not in this slide yet. So if evolution is not happening, then mating is random. There's no, even in a human population, mating is fairly random, even though you as an individual decide who you mate with. But mating is random, meaning that you're not taking certain genotypes or alleles out of the population, preventing somebody from mating with another trait. And so none of the mechanisms of evolution are happening. It's a consistent equilibrium population. And okay, so that's the thing that we found. We figured out that our P was 80% or 0.8 and our Q is 0.2. So if this is if our flowers or our red and our white alleles from our flowers are represented by these little balls, 80% is red and 20% of the genes in the population are white, then that's kind of what it would look like inside of a little container, right? And if you were to take, we don't know if these are male or female, but if you were to take them out, each female version would have 80%, should have 80% chance of giving you the red and a 20% of the white gene. And same thing for dad. You have equal chances with each of those. And it should remain consistent. We shouldn't really see much change around the red coloration being 80% and the white coloration being 20%, or the white allele. So that tells you that P plus Q equals 1. That tells you the gene, the percentage of that the gene is in the population. But to figure out what the percentage is, the frequencies or percentage of how things look, the phenotype, you would use the equation there. And that's the true Hardy-Weinberg equation. P squared plus 2PQ plus Q squared equals 1. And we figured out already what P is and what Q is. And so we can just plug it all in to figure out what the frequency would be of red, pink, or white looking flowers based on the genes that we know are in the population, which we already knew before because we had that. They showed us that we had like 320 and 20 pink ones or 20 white ones and 300 and something of the red ones. So we're just kind of going in a big circle. So knowing that 80% of the population is, or 80% of the population has the red gene, then you can, P squared is 0.8 squared, 0.64, 2PQ, right? 0.8 times 0.2 times 2, and then Q squared, 0.2 squared. And if we add all that up, it should equal 1. And if it didn't, then we did our math wrong. And we're not doing Punnett square in this, so I'm not really going to look into that. So on the exam, you might be told what your P is of a population. Well, now you should be able to figure out what your Q is also. Or maybe you're just given numbers. You're just told that in a population of 1,000 individuals, there are 800 of this gene. Well, of the dominant gene. Well, you can now know just by subtracting from 1,000, what is your Q. So just be able to, if you're given one thing, know how you could answer. I'm going to close this door. Know how you could answer, how you could tell me the phenotype, how you could also tell the allelic frequency or the genes that are in the population. I feel like I made that incredibly confusing. Does that make sense? Do you want me to go over that again, or do y'all feel like you have it?
I think we got it.
I
I got it.
Okay, good. Are you okay, Paula?
Yeah,
Okay.
I had a train of thought and I lost it, but
There
yeah,
are
because I
there
guess
are
the difference between is it a difference between the frequency and the phenotype? Are they two different equations?
yes. So there's allelic frequency and phenotype frequency. So
The
allelic
allele
frequency,
frequency is the 2,
yes.
where we were multiplying the 320 by the 2
Uh,
from the one before when we were figuring it out.
the it's just P plus Q equals 1 is your
Okay.
allelic frequency.
That's my, okay, the P, okay. So then the Harry Weinberg were I guess the frequency, this one here is the frequencies equation
Of your phenotype,
is my
yes.
phenotype.
And
Okay.
the way I remember that is because if you look at it, I mean, it makes sense, right? If you're homozygous, you have 2 of 1. 2, you have to be homozygous dominant, homozygous recessive. And then this guy in the middle, you have P and Q, you're heterozygous. And so you might have, if you're looking at the red, the white, and then a pink flower, you have 3 different looks. That's kind of how I remember that this is the phenotype.
Okay.
The genotype, you can only get one or the other from mom or dad.
Okay.
And if you got two different ones from mom and dad, it makes you heterozygous, but you still only have the option of getting one or the other from mom or dad. Okay. What is that? Okay. So in true populations, we are rarely in Hardy-Weinberg equilibrium because frequencies are changing all the time due to evolution and other situations like environmental situations and geographical barriers that aren't necessarily controllable. So in order for Hardy-Weinberg to be met, to be in equilibrium, there's things that have to be happening. There has to be no mutations, which happens all the time, even in a human population. There are mutations to our genes. Mating has to be random. So you can choose who you want to mate with, but it's still random mating. It's not like there's a law that says you cannot mate with curly haired people. That would be non-random mating. There's no natural selection occurring. The population must be very large. Small populations are never in Hardy-Weinberg, and there cannot be gene flow. And so we'll talk about what gene flow is and some of these other things in just a second. That's just an example that I don't necessarily want to go into. So 3 factors that the 3 main factors. So I would know the 5 things that have to be happening in order for Hardy-Weinberg to be in existence. And it rarely is. There's always changes in allelic frequencies or genes, the percentage of different genes in a population. And there's 3 things that will make that change occur: natural selection, genetic drift, and gene flow. So natural selection is differential success in reproduction. So in this example, we've got a bunch of mosquitoes, and back in the 60s when we were using DDT, which was a very powerful pesticide, but a very effective pesticide, it was overused and we had insect resistance to it. DDT was used in farms. It was used, in this example, it was used to control mosquito populations. And so we spray DDT, and all of these blue mosquitoes die. They are susceptible, they get sick, and they die from the pesticide. But there's a couple handful in the population that don't die for whatever reason. They're resistant to the pesticide. It doesn't affect them in some way. And so these two are left to continue to breed and pass whatever that gene is on to their offspring. And over time, this is a very truncated version of it. There should be steps in between where you still have a few blue ones left. But over time, you have selected for only mosquitoes that are resistant to this pesticide, because you killed off all the other ones that died from the pesticide. So that's an example of natural selection that would be brought on really by human use of pesticides. So you select for something. We're selecting for mosquitoes that don't die from the pesticide by applying and applying and applying. And they only are who is left to continue to pass on those genes. Genetic drift describes how the allelic frequencies fluctuate. Genetic drift tends to reduce genetic variation because we lose alleles, and this tends to occur more in smaller populations. So in this case, the highlighted plants are the only ones that make seeds and are pollinated for whatever reason, and make more of themselves, make offspring. And so only 5 of 10 plants leave offspring. Well, in this case, now it's only 2 that are going to start to leave offspring. And as over here, we had pink, red, yellow, and a couple pinks. Well, for whatever reason, now we only have red ones that are left, and they're only going to produce red ones over time. So we start to see this gene flow where you're only one color, one phenotype is left behind. Well, that's not a good example. I'm trying to think if I can imagine in my mind another one. Genetic drift describes how allele frequencies fluctuate unpredictably. So it's just unpredictable. We were very diverse, a little bit diverse, and then the next generation we were not, with little to no explanation for why that happens. Founder effect is when you have a few individuals that get isolated from the population. So this is usually like an earthquake happens, and you have like a vault or a hole that happens in the earth, and you separate 2 populations, and the big moose population of hundreds of moose are over here still living, and there's 2 that are left behind. And it's like inbreeding almost. So you have less genetic makeup to start a new population, and maybe these 2 moose populations become very different and isolated from one another. So bottleneck effect is one example of, founder effect is few individuals become isolated from a large population, start to reproduce. Bottleneck effect is there's some drastic reduction in the population size. Generally, it's due to an environmental change. So there's a volcano and it burns up most of the birds before they're able to fly up in the air. And so you went from a large population and a bottleneck down to just a few, and you've reduced the genetic availability in the population. And then just because it's randomly, the resulting gene pool might not look like the original population's gene pool, because it's random who survived and who didn't. So bottleneck population, I guess this Florida panther went through that from, I don't know if it was a geographical environmental condition, or if it was over, hunting by people, but your original population has a lot of genetic variability in it. And then with bottleneck effect, only a few survive, and you end up with majority of blue balls and only one little white ball, but we lost all these little golden yellow colored ones. And so the surviving population looks very different genetic wise than what was originally in the population. This was another case study of the prairie chicken, but I'm going to go through this because I don't want to keep y'all forever and ever. So a summary of genetic drift, it's significant in small populations. Okay, so genetic drift is really significant in small populations. Genetic drift is just the movement of genes. There was a slide in here that I thought had a definition, but I guess not. Genetic drift is just the movement of genes from over time. It's significant in small populations because you have less to dilute it, right? If we had a human population that was very, very small, then, or was isolated, like if you think of the island of Japan, right? Everyone looks very similar. But in larger continents, Russia, United States, North and Central and South America, um, you can't always tell where people came from or what their ethnicity necessarily is, or their culture, or their background, because those are giant populations. And so we're just this big melting pot. But in small populations, genetic drift is much more significant. You see those changes. Drift, movement of genes, can cause allele frequencies to change randomly. And it can lead to either genetic loss, the loss of genetic variation, and it can be harmful. Loss of genetic variation, which can be harmful, but it can also cause those harmful alleles to be fixed because they've been removed from the population. So gene flow is the movement of alleles among populations. They can be transferred through movement, so pollen through wind pollination, or from individuals. We move our gametes, right? As we mate, it leaves the man and goes into the woman. Gene flow tends to reduce genetic variation among populations over time. I don't know if I want to do this one. Oh, this is like these little birds. So I'm trying to remember the story behind them and I can't really recall it. Immigration of birds from the mainland introduced alleles that decrease the fitness of the island population. So birds from the mainland started to come into the island. So that's the drift of genes to the island. And it actually, by introducing those genes, it was detrimental because it decreased their fitness. They weren't tough enough to live on that island for whatever reason. It can also sometimes increase the fitness of the population. So another example using mosquitoes and West Nile, when they become resistant to the insecticides, that increases their fitness because they're surviving better. Bad for humans, good for the mosquitoes. So sometimes gene flow can be a good thing. Okay. Main thing with that is understanding gene flow. Let me go back to the concepts. What natural selection is, what genetic drift is, and what gene flow is, and founder effect versus bottleneck effect in genetic drift. And then the bigger one is understanding that Hardy-Weinberg equation, the P plus Q equals 1, P squared plus 2P plus 2PQ equals Q plus Q squared equals 1. Okay, are y'all good with that one? I know it's like 7:18. I do want to get through chapter 22 for you because I think there's some concepts to go over on that one. And then we can probably call it quits because 23, I think, was just regurgitation, and 26, you guys already covered in lab. 23. No, that's not what I want to look at. Hold on. 21, 23, 22. So if I remember right, the origin of species. So talking about speciation and how species are made. And then some of those post-zygotic and zygotic, pre-zygotic barriers. Galapagos Islands again. So speciation is the process by which one species splits and becomes two or more species. What makes that happen? And there's two different concepts to that: microevolution and macroevolution. So macroevolution, macro is big. And this is like, big changes in species, right? Like how a common ancestor was a cell, and now we have that Neanderthal, whatever he is. And then microevolution, these are all separate species that have a shared common ancestor. But even to someone who isn't necessarily scientific or studies canines, that they look very similar. And you would guess, even uneducated in phylogenetics or biology, you could probably guess that they are more closely related to one another than to birds, right? So microevolution is changes in allelic, microevolution consists of changes in allele frequency in a population over time, where macroevolution refers to broad patterns of evolutionary change above the species level. Okay, the biological species concept is a way to understand, one way to explain to ourselves how speciation occurs and how species are made. So the biological species concept says that to be a species and a separate species, you must be a group of a population whose members can interbreed with one another in nature. So you can't be a species necessarily if in the nature that you're in, you can't interact with one another. You can't come together. You live on different continents, separate islands. You can be considered separate species. So you have to interbreed in nature, and the offspring that you produce must be viable and fertile. So an example is a horse and a donkey produces a mule. While it produced offspring, the offspring is not fertile. So that is not, those are not the same species. Donkeys and horses are different species because when they mate, their offspring cannot reproduce themselves. And between species, different species, you can have very close similarities. Those two birds are different species. And then looking at humans, we're all incredibly diverse, but we are the same species. We can interbreed with one another. Reproductive isolation is when there's a biological barrier that impedes two species from producing viable, fertile offspring. Sometimes you can get hybrids, like horse donkey makes a mule. But those hybrids generally die off very quickly, or they aren't unable to reproduce themselves. So prezygotic barriers to speciation is a zygote. When egg and sperm fuse, when they fuse and become one cell, that's now a zygote, right? So prezygotic is they're not even able to mate. It's before mating occurs. So there's a block in fertilization. Fertilization cannot happen. It keeps different species from mating. It prevents the successful completion of mating, or hinders fertilization even if the mating process can happen. So there's different things that can happen, like habitat isolation. They're not located anywhere close to one another to be able to mate with one another. Temporal isolation could be one is active at night and one's active during the day. One goes to another location when it's winter time, or they migrate to different places, and so they never overlap. And then behavioral isolation would be more so the nocturnal versus diurnal. I live in the water, you live on land, kind of a thing. And so mating never occurs in that way. So habitat isolation, two species encounter each other rarely or not at all because they're in totally different habitats, tree versus the ground. And it's not necessarily a physical barrier like a mountain that separates them. It's that they choose different places. If you force them to mate, they probably could. Temporal isolation, they breed at separate times of the day or different seasons, or different years, and cannot mix their gametes. So some, not necessarily animals necessarily, but a lot of plants will not reproduce, but like every other year. And so maybe those years are not overlapping. And then behavioral isolation would be courtship rituals and other behaviors that are effective barriers, such as I swim, you stay on land, that kind of thing. I'm only attracted to you if you do this special little courtship thing. So those are prezygotic barriers. Mating never happens. Nothing can occur. Postzygotic, where are we looking at? Mechanical isolation, gametic isolation. So more, I guess, in another prezygotic barrier, mating can be attempted. The ones that we were talking about, mating doesn't happen, but fertilization cannot take place. So there might be morphological differences. So lock and key mechanism, the male part doesn't fit into the female part. They can't mate with one another because they don't fit together properly. Kind of like you hear about how pigs have like they're a corkscrew, and so the female has to be able to accept that corkscrew. Well, so a pig can't mate with a dog or something because it can't accept that. Gametic isolation, gametes being the genes, the sperm of one species just can't fertilize the egg of another species. So maybe a dog and a cat could mate with one another, but the dog's sperm cannot fertilize the cat's eggs. Post-zygotic barriers happen after mating has occurred. So they are able to mate, they are able to fertilize the egg, but the resulting hybrid is that the viability is low, so it's not able to live very long or to make it itself to the point where it can reproduce. The hybrid is infertile, or it has very reduced fertility, or the hybrid just starts to decay and break down. It never really makes it. So, I guess I don't know what this little lizard is, but that's reduced viability. The donkey and the horse making the mule. There are cases when mules are able to reproduce, but it's incredibly rare. So most of the time mules are sterile. And this is an example of wheat. And so the hybrid starts to break down. The hybrid is teeny tiny. It's just not, it doesn't produce whatever we get from wheat. Genes of different parent species interact, but they impair the hybrid's development or survival. And I don't know what animal that is, to give you an example. It's talked about sterility. And then first generation hybrids are fertile, but when they mate with others, the resulting generations just are worse and worse and worse. And you get these terrible, so this might be a parent, or this might be the first hybrid. The second generation is smaller, doesn't produce what it should. The third generation is just really puny. So pre-zygotic barriers, mating either can't happen, mating does happen, but fertilization does not. Post-zygotic barriers are when mating can occur, but the viability, fertility of the offspring is way puny. So biological species concept emphasizes the absence of gene flow, meaning that we're able to make random hybrids of things. So you could make these things called growler bears, where you artificially, or you force or put them together, a polar bear and a grizzly bear in the same location, and they produce a viable, fertile offspring. But they're not, that's never going to happen. And so we can artificially make two things appear to be the same species because they're able to produce those offspring, but it's not going to happen naturally. Same thing with tigers and lions. You can make a liger, but they're on different continents. It's not going to occur in nature, in nature and in the same time. The morphological species concept is a different way of defining species, and it looks at structural features. So biological is that you reproduce, you produce viable offspring. Morphological is that you look very similar to one another. And then ecological species concept views the species in terms of its ecological niche. Do they live in the same space? Do kind of the same things? These are less popular definitions of species, but sometimes you have to use all three to make a decision if two things are truly different species, or if they could be considered the same species. And phylogenetic species concept defines the species as the smallest group of individuals that share a common ancestors. They branch off in that phylogenetic tree. And these, so phylogenetic species and ecological species and morphological species concept helps us choose things that are asexual and decide if they're species or not. Biological species, you have to be a sexual species. You have to be able to mate, reproduce. Asexual is where you just produce clones of yourself. And how do you define that, right? How do you define if they're separate species or not? Well, you use one of these three species concepts. Speciation can also occur in two different ways: allopatric speciation and sympatric speciation. Allopatric is when gene flow is interrupted because there's a geographical barrier that isolates two populations. So, oh, I see. So something happens here where you have, oh, I see. So you have this little population and this growth happens in the ground. The soil starts to build up, or the water goes down, I guess. And you separate these two fish and you end up over time with two different species. Or the water goes up, and so the fish are now able to mate with one another. And so gene flow changes over time because of that geographical barrier or geographical isolation. Definition of geographic barrier depends on the ability of the population to disperse. So a canyon could be a barrier for rodents, right? That's a huge place for them to go, but it's not going to be for birds, right? Because a bird can just fly over the canyon. May not necessarily be for a larger animal like a coyote that travels long distances, or a wolf, and definitely not for pollen where the wind is going to carry that. So a geographical barrier is going to depend on the individual species. I'm going to skip through this one too, I think. And then sympatric speciation takes place in populations that live in the same geographic area. So this is when gene flow is reduced between groups that remain in contact through factors that include polyploidy, habitat differentiation, I like the tree, you take the ground, or sexual selection. So polyploidy is just the presence of extra sets of chromosomes. It's just random. It's like mutation almost in a way. Not really common in animals, because when it happens in animals, we think of trisomy 21. It usually is kind of to the detriment of the individual, but in plants, it's incredibly common. And it happens during cell division. Accidents happen. You end up with extra chromosomes than you should. An autopolyploid is an individual with more than two sets of chromosomes. I don't think I care that you know what that is. And an allopolyploid is a species with multiple sets of chromosomes from different species. I don't care so much that you know what those are. Just know what that a polyploid, you have extra chromosomes. That's going to affect genetic drift. And habitat differentiation. So you have different ecological niches. So like a maggot fly can feed on hawthorn trees as well as introduced apple trees. They use different host species. Flies that use different host species are reproductively isolated by habitat and temporal barriers. I think the easier way to think of it with habitat differentiation is location of where you like to feed and live. An arboreal or a tree dwelling animal versus terrestrial or on the ground. And then sexual selection can also drive sympatric speciation. And so that's like where you look for something specific in your mate. So peacocks want to see, the female wants to see bright colors. If it's an old guy or it's a puny guy that's lost all of his tail feathers, they're not going to be attracted to it. Or in the case of fish, cichlids especially, they mate, different colors they're only going to mate with individuals of certain colors. And so that makes them separate species, but probably they could all have the ability to mate with one another if they were forced to. So allopatric speciation is geographic isolation. It restricts the gene flow. And sympatric speciation is there's some reproductive barrier, habitat, sexual selection, and what was the third one? The first one that we talked about? I already forgot. Oh, polyploidy. You just have random extra genes. Oh, it was right there. I could have just read the second thing there. Okay. So we're a little bit over an hour. I'm going to stop recording. Do you guys have any questions on those?